Yes, good evening to everyone. So, welcome to day two session on target modeling and validation in internship and certificate course in AI and ML approaches of drug and vaccine design. So, before stepping on to this today's session, can we recap the last session topics? What exactly did we discuss in the last session? So, do you remember the last session topics? Yeah, either unmute and answer or else you can type the answers in the chat box. So each and every day, the first 5 to 10 minutes, we discuss on the last session contents. Very good. So Atharva Patel has initiated that we have discussed on various biological databases like PubChem, CBioportal. Very good. So PubChem is related to the chemical database. That is, if you want to download 1D, 2D, 3D, we can able to download the structures from PubChem. And CBioportal is for genome database, like cancer genome database. If you want to get any information related to cancer, any particular type of a cancer, we can get this information. So one is chemical, the another is related to the gene and the protein targets. So we have discussed majorly on databases, isn't it? So on yesterday we discussed on biological databases. Under biological databases, we have discussed sequence, structures, and we have also discussed on the protein-protein interactions. Yes, that's what. Okay, so protein-protein interaction database, and then we have discussed on disease database, isn't it? You remember the disease database? So give me some examples for disease database. So what are the databases exclusively we search for disease information? Of course, CBioportal is related to cancer genome. Like this, we have so many. Yes, OMAM, very good. This is what I anticipated. And even KEGG, Kyoto Encyclopedia of Genes and Genomes, has disease pathway exclusively. Disease pathway is a separate database present in KIG. Similarly, if you are working on inheritance, that is a genetic and genetic disorders and gene-related diseases, then obviously we have to work on OMIM. So what is OMIM? So I think you know the expansion of OMIM. Can you expand OMIE? Did you hear about OMIE? What is OMIE? So if you know the expansion of OMIM, you can able to know this expansion. Yes, online Mendelian inheritance meant obviously then that is inheritance in animals. Online Mendelian inheritance in animals. Okay, very good. So this is what, okay, we have seen a few things and we have excluded something, but we can discuss during the recap of the last sessions. And give me some examples for PPI databases. PPI databases. Funko, intact, super. How about the others? Very good. String, TPI, database of protein interactions. Good. So I think you mentioned the major kind of the PPI databases. I've classified three types of databases. You remember? So three types of data. Of course, BioGrid is also one of the protein-protein interaction database. So there are three types of databases we have discussed on yesterday, isn't it? Out of these three types, what are the examples I provided for secondary databases? Examples for secondary databases. Cut SCOP, structural classification of proteins. Uniprot won't come under secondary. It comes under primary. Cath, very good. Class, architecture, topology, homology. Of course, motif, okay, and profiles, fingerprints. So something which we didn't discuss on yesterday, and giving few more additional information. Profiles, fingerprints, motifs, domains. So these are some of the secondary databases. Okay. So anyhow, we'll be discussing on the tertiary and other databases while going for a demo. Now, let me go ahead on today's session. So today's session we'll be discussing on target modeling and validation. Of course, this is also a voluminous chapter. So before that, I just providing you some information about Sri Venkateswara College of Engineering. So actually, the PhD scholars receive monthly stipend of rupees 18K per month for the first year, and for the second and third year they'll be getting 20K. So she is my PhD scholar. She is working on the effect of phyllanthus neruri compounds on heat shock protein 90 in two types of diseases, that is type 2 diabetes mellitus associated with NAFLD. Okay, so this is the research fellowship. And now let me go to this contents, today's session contents. So we'll be discussing on target discovery, target identification, target deconvolution. So what is the difference between these three terms? And then we discuss target verification, validation. Then we discuss on databases and network pharmacology, target identification databases, and we discuss protein structure prediction, etc. Now let me go ahead with one by one. Target identification. What is target? Actually, if you see in drug design, especially we have two terms. One is ligand, the another is target or a receptor. Okay, so whatever we call it as a receptor is what we call it as a target. So generally, if you see these two important terms in computer-aided drug design, okay, the ligand is usually a drug. Okay, it's a small molecules. Ligand generally you have to remember it is a small molecule, and receptor is a macromolecule. So the in receptor you will majorly find protein as the target, protein as the target, or also we protein as the receptor. And if you see the ligand can be a drug, a small molecule, or it can be a peptide, or it can be DNA, RNA, lipid, or carbohydrate molecules, or even it can be a protein itself. In antigen antibody interaction, whatever molecule is smaller in size, that will be considered as a ligand. So that kind of a protein, the protein ligand generally binds with the protein receptor, even in signal transduction in most of the immune proteins. So there also you can find like this. But one thing you have to remember, the ligand is a small molecule, receptor is a large molecule, and the other name of receptor is target. More about this we'll be discussing what the other kind of ligands and receptors we'll be discussing in the subsequent sessions. So, what is target? It is a molecular recognition site to which drug binds. So, generally, if you take a simple term like a drug and protein, so the target is what we anticipate the binding sites. The recognition site is what we call it as a binding site. And if it is a normal protein, we call it as binding. But if it is an enzymatic protein, we call it as the active site. We call this as an active site. Okay. So for normal protein, I'm telling you.
உனக்கு தான் இன்டர்ன்ஷிப் எங்களுக்குலாம் இல்ல.
Okay. So if it is protein, okay, it is we call it as a binding site, and if it is an enzyme, we call it as an active site. Okay. And if you see the target molecule, it is a receptor, enzyme. That's what we have discussed. Transport molecule, ion chain, channel, tubulin, immunophilin. We'll be discussing more about this. Strategies for identifying drug targets. There are two strategies for identifying drug targets. Number one is the conventional strategies. Under conventional strategy, we can either analyze the pathophysiology or we can analyze the analysis of the existing drugs. We generally have to understand pharmacopoeia, pharmacopoeia method of analysis. MOA stands for the method of analysis. So one should know how the drugs are synthesized and what are the methods used, what are the different methods, and how about the yield. Okay, all these things are coming under a kind of a method of analysis in a pharmaceutical industries. They have a kind of a standards for preparing the APA. Generally, whatever we call drugs, they call it as an APA, active pharmaceutical ingredient. In new strategies, we generally go with identifying the disease genes, and we go with the disease modifying genes. So in new strategies are majorly targeted for precision medicine. If you want, not all the drugs are going to treat the diseases. Okay, whatever drugs specifically mentioned to treat a disease, that is that can able to treat only few patients, not all the patients. If you want to target all the patients, obviously we have to use this kind of a new strategies. One should know about this information. And generally, if we see the targets, the major targets or receptors which accounts for around 45 percentage, and next to that, you can see enzymes, which accounts around 28 percentage. Okay, so this is general statistics taken on the targets, the types of targets for different diseases. And next to that, enzymes, you can see hormones and the other things. Now, let me take you to the distinguish of target deconvolution as well as target discovery. What is target deconvolution? So if you see the target deconvolution, okay, first we should have a compound. That is, if you have a 1D, 2D, or 3D structure of a compound, you can feed this as an input in the target deconvolution software. So what it does, it's going to provide us a series of target receptors. So, and whatever it is going to bind, whatever chemical compound we fed as an input, and whatever it's going to bind with the different receptors, we also get a kind of a score. Based on the kind of a score, we can able to find the ligand binding affinity with respect to the target receptors. Okay. So first we should have an input of a chemical compound. Even 1D information is sufficient in this case. And if you see, the next thing is a target discovery. Generally, we want a compound. So we have plenty of compounds in the databases, and we have a target. And so if we want to get which is the best compound binding to a particular target, okay, then that's what generally we come across in target discovery. So generally in target discovery, if you're focusing on multiple compounds and if you want to screen a best compound, the process what we use in drug design is called virtual screening. So try to screen a best compound from series of compounds or related compounds, okay, by binding with one particular target receptor. So if you see in both the cases, okay, it can be a target deconvolution or target discovery. Okay, we have to focus only on a single target. So what is the necessity to go with the multiple targets? Even if we get multiple targets, we can get some idea like on whether it's what type of inhibitor compound is that. Okay, so if you want to know about the type of inhibitor, that you can able to get from this target deconvolution information. But obviously, one should end with screening a single compound, and that should be targeted with a single protein target. Okay, so this is what we distinguish target deconvolution and target discovery. Now, let me go ahead with this protein structure prediction. So, what is the five levels of protein structure? You know, actually, I mentioned only four levels of protein structure. One level of protein structure is missing here. Can you identify and tell me by unmuting, or you can type the same in chat box? I have shown you four levels of protein structure, but I am mentioning you that we have five levels. I missed one level. What is the missed level? What is that I am claiming for? Do you want me to show once again? Can anyone tell me the missed level of protein structure prediction? No? Okay, then I can take you to the slide itself. See, the first thing what we see is the primary structure. The primary structure of a protein is an amino acid sequence. The second type is the secondary structure. Here we have alpha helices. The alpha helices, we have types of alpha helices, like the alpha, alpha 3 alpha 10, and we have pi alpha. We have types of alpha helices, and similarly in beta sheets we have parallel, we have anti-parallel beta sheets, loops, turns, coils. This comes under the secondary structure. And the third one, whatever I missed, is super secondary. Under super secondary, how you find the protein structure? The protein structure are generally present in the form of motif. Motif. Okay, so I think now we can remember the examples of motif. The examples of motif like beta, alpha, beta, Greek key motif. So these are the types of motifs which comes under super secondary structure. Why we call this as super secondary structure? The super secondary structure, we have a combination of beta and alpha helices, okay, alpha helices and beta sheets in this case. And in the Greek, you can find the beta sheets in combination. So just because we have this combination of secondary structures, we call this as super secondary structures. And secondary, obviously, the name implies the alpha helices is a combination of few amino acids. Okay, it have nearly 5 to 15 amino acids. Okay, that's the base. We also have two types of alpha helices based on the length, like short alpha helices, long alpha helices. The short is just 5 amino acid in length, and the long can extend even up to the 15 amino acids. So it like this, you have a combination of the secondary as a super secondary, and the fourth type is the tertiary. In the tertiary, we obviously have a combination of super secondary motif. In this case, we call this structure as domains. Okay, so and if you see in the structural term of the protein, we call this as chains. Domains are also called as chains. Okay, so we have only one chain. We have only one chain in the tertiary structure, and of course, it's a pack of the super secondary structure. But whereas if you come across the final level of protein structure, that is quaternary. In quaternary, we have multiple chains. In this case, you can see the dark region of alpha helices. The alpha helices are shown in this kind of a cylinder. Okay, and beta sheets, the beta sheets are shown in the form of an arrow mark. Okay, so this is how a beta sheet looks like. If alpha is looking like this, which is going to have an N terminal followed by C terminal, and the beta sheet is similarly going to have N terminal and C terminal at the top and N terminal at the bottom. This is how the alpha and beta sheets looks like. And here you can see a pack of alpha helices, and one color, whatever we see, if you name it as A chain, and this light color can be a P chain, and in between you have a C chain and D chain. So because we can see four different colors, here we can see in the quaternary structure, it is a pack of four chains. Okay, so this is how we can see the levels of protein structure. And if you see the composition of protein, so everyone know amino acids are the building blocks of the protein. But if I ask me, I can tell you this is not the major composition. There is one more molecule which comprises majorly in protein. Can you tell me, other than amino acids, what is the major molecule we find in protein? But no doubt, amino acids are the building blocks of proteins. Is good. Super secondary, okay. But the next question is, other than amino acids, what is the major molecule you can find? Can I give you the answer? Water. Even in our body, more than 70 percentage of the fluid is nothing but water. So here you can see that. But if you see this first building blocks, the amino acids with electrically charged. So we can have positively charged amino acid, negatively charged amino acid. We have amino acids with polar uncharged side chains, and similarly we have amino acids with the hydrophobic side chains. And we also have special cases like cysteine, glycine, and proline. Now, let me go ahead with the peptide bond. What is peptide bond? A peptide bond is a combination of two different amino acids. Generally, the two different amino acids join through peptide bond. During the formation of peptide bond, whatever you can find here, okay, so whatever peptide bond is formed between a merging of two different amino acids, a water molecule is released. The OHH molecule is going to release in the form of a water molecule. Now let me take you to the secondary structure of a peptide. So if you see the secondary structure, okay, so one should know about the angles like phi, psi, omega. So what exactly is phi? The phi is between n and c alpha. Okay, if that is phi, then c alpha and c double bond O. C double bond O is what we call it as psi. And obviously C double bond O and N. Okay, we call this as, or I can simply tell like C alpha and C. No need to show even this double bond O. And similarly, C and N is what? Omega. So if you see in this types of secondary structures, alpha helix has a different phi psi omega angle. Similarly, whatever alpha helix I mentioned, this is defaultly a right alpha helix. Similarly, we have left alpha helix, retail helix, pi helix. Okay, so for all these helices, up to this or alpha helices, okay, and up to this or beta sheets. And if you see each and every secondary structures have a different pi, psi, and omega angles. And next we come across the secondary structure of alpha helix. So one should know this structural proteomics. Okay, so to work extensively on precision based drug design. Okay, so I'm giving you some more fundamentals related to the secondary structure. If you see the secondary structure of alpha helix, the most favoring amino acids of alpha helix are methionine, alanine, arginine, and lysine and leucine. If these are most favoring amino acids for alpha helices, then the worst formers, okay, unfavorable amino acids or proline, glycine. You know why we have these amino acids as an unfavorable amino acids for the alpha. So the reason behind the proline, in proline we have a single restricted rotation. So we don't have all these three angle rotation in the peptide bond formation. We have only one kind of a rotation, that to a restricted rotation of phi in proline. But whereas if you take the glycine, the singlet amino acid is G, we have all these three angle rotations, that too we have flexible rotations. Just because we have all these three angle rotations, that too flexible amino acid. So we can't able to have these amino acids in these turns. If you see the alpha helix, okay, so we can see this kind of an alpha helix. So for each and every three amino acids, or each and every 3.6 residues per turn, you can able to see in a default alpha helix. In default alpha helix is generally 3.6 residues. So this is one turn. Here you can see only 3.6 amino acids. Okay, and if you see in these turns, we can't anticipate these. These are not welcome proline, because proline, restricted rotation, so we can't have, and even glycine, because of flexible rotation, the alpha helix can break at any time. So we have only the 5 amino acids. Other than these amino acids, we also have the propensity for the other amino acids. Okay, based on the propensity, alpha helix propensity, one can able to find whether the sequence of amino acids of 5 to 15. I told you, no, that is the preferred size of alpha. If so, what exactly is going to find, what is exactly is going to form alpha, what exactly is going to form beta, is just dependent on the amino acids present in the sequence. Okay, even if you want to develop any algorithm for secondary structure prediction, first we have to know the amino acids. We have to give the propensity of alpha helix amino acid, and then we have to predict the beta sheet. Whatever is not going to form the alpha and beta, it's going to form a coil or loop or turn. So these are the other secondary structure elements. Okay, and now let me go ahead with the next information on globin family. So if you see in this example, okay, I'm mentioning you about the globin family. So hemoglobin A chain, hemoglobin B chain. Okay, so whatever I mentioned, like hemoglobin is from the human source, Homo sapiens. And we are comparing with the myoglobin of swine. Okay, myoglobin, that is muscle protein of swine. Okay, and we can or also comparing with the leguminous hemoglobin. Leguminous swine, I think it's a pig. Leguminous. What do you mean by legume? It is a pea plant, isn't it? That is a pea family, chickpea family. So, if you compare the human and animal and compared with the plant, now next thing is the bacillus kind of hemoglobin, sea urchin hemoglobin. So, if you compare the different kind of a species, all these globin fold proteins have most similar structure. You can't distinguish the structure of the globin protein of different species. But if you compare the sequences, you find variation with respect to this sequences. That is, if you compare the homologic comparison with the amino acid sequence, the sequence can give a homology, that is percentage homology from 99 percentage to 16 percentage. Even human hemoglobin A chain is just having 99% similarity with hemoglobin of P chain. But if you compare this hemoglobin of A chain with the chimpanzee hemoglobin of A chain, you can find around 100% similarity. So it depends, okay, not just with respect to the species, I mean, not with respect to the chains, but also with respect to the species. Okay, there are some the close ancestors have a more closely resembling sequences in comparison with homology. So this thing you should understand. Even though structure is same, the sequence variation is there. The next, how this 3D structure of proteins structures are formed. The well-known, I told you on yesterday itself, X-ray crystallography is the gold standard technique used to get the protein structure in the form of PDB file. Okay, how to get first of all protein structure? We have to get a protein crystal. Okay, getting a protein crystal is not an easy task. If you see, if you want to crystal even a small molecule, small compound, a chemical compound drug, that itself we have to have much of chemistry knowledge. One should know about unit lattices, etc. Anything you want to clarify in between? What no one has mentioned. Any doubts? Anything? Then
Yes, sir. I do have one doubt. If
yeah,
you want
please.
permission, sir. While um, doing the docking for protein and ligands,
Mhm.
do we have to take care of alpha, psi, as well as gap, whatever the angles you told us, so that there is a proper matching? And another doubt is how will we know that whether the docking has done perfectly well or not? Because sometimes it happens that it becomes very much this one poisonous or something like that. So
Good, good.
that is
Actually, these are the kind of uh, doubts I'm anticipating from your side, but this is not the right time. Actually, we have this molecular docking session separately on session 5 and molecular docking analysis virtual screening methods on session 6. So whatever
okay,
question
sir.
you have asked, okay, it's related to that kind of a sessions. Don't forget to ask these kind of a questions when I'm taking on molecular docking. Okay.
Not an issue, sir.
Uh,
Thank you very much. Thank you
yeah,
very
yeah,
much.
because even a few participants, I don't think they know few fundamentals. Let me complete these fundamentals, and if I come to the unit 5, definitely I clarify everything. Okay, now let me go ahead.
Okay, sir.
Good, thank you. And crystal, whatever we get the protein crystal, it's very difficult because it's a macromolecule. We generally measure the proteins in 10 kilodaltons to 100 kilodaltons. If the protein size is much more, up to kilodaltons, I told you it's like a 10K to 100 ADA. Okay, it's a challenging task, isn't it, to bring in the form of a crystal? Because even the chemical compounds itself, you want to purify through the crystallization process, and if you want to bring some crystals are not formed in the magma, But if you can able to make it, okay, that's a most important and difficulty task. If you can able to make it, so this we can take it as an input sample, and then we can go with the analyzing the diffraction pattern. If we pass the X-rays even in our human body, only the bones are getting reflected, isn't it? The black, white, and gray matter. Only these three colors we can able to find. So similarly for the protein structures, we find these three, black and gray colors in the white background. So whatever we find these, okay, each and every dot is an amino acid, and based on the intensity, even the amino acids got determined, but practically we can't able to make it. So one thing we can able to make it using the simulation. So using a simulation, we can able to convert the electron density maps. The electron density maps or the intermediates were through the we can able to generate from the softwares from the diffraction pattern. So whatever the electron density maps we have to simulate it to get in the form of a PDB file, which is an atomic model. Okay, so this kind of a refinement we also happens in the form of a cycle until we get a kind of a structure, a clear structure. We do this kind of a process. And a part of a question, whatever the participant earlier asked, okay, sometimes I'll be answering to that kind of a part of the question, whatever the participant recently asked. Like sometimes the crystals are not formed perfectly. Okay, so a few amino acids might be missing. Okay, for example, out of 732 amino acids, maybe first 1 to 26 amino acids will be missing, and we can able to see the this kind of amino acid like 27 to 742. Okay, if the 742 is the total size, so this size of the protein is finally developed from the X-ray crystallography. It means that we have some missing residues in the protein. So this is not a fault with us. Actually, this is a fault with the crystallography department who has brought that kind of a crystals. Okay, to so whatever they got a kind of a crystal, okay, they may find this kind of a missing residues, and only whatever data available from the data, only these kind of a protein is modeled using this even gold standard techniques. So this kind of a missing residues won't hinder you during the docking process. And if these amino acids are not significant with respect to binding site and active site, we can simply ignore. But during the simulation, we have to add all these amino acids, missing residues. Now let me go ahead with the tertiary structure prediction. So in the tertiary structure prediction, okay, we have three methods of tertiary structure prediction. So homology modeling, threading, and ab initio. In homology modeling, we have these tools, and if you want to work on any of these tools, one can go through this URL called xpassi.ch/tools. We have plenty of tools to work with the truck design, especially to work with the truck, target. Uh, then we can use this kind of XPASI web server. In homology modeling, the well-known tool what we can use, online tool we can use is the Swiss model, which uses a automated knowledge-based protein modeling server. Similarly, in my lab, we are using the Fiery for threading. Okay, protein homology analogy recognition engine. Protein homology analogy recognition engine. So this is a tool for threading. So what is the difference between homology modeling, threading, ab initio? We'll be discussing in the subsequent slides. And one can able to check these are the tools of the ab initio. I have already used to what if ProCheck, ProSA Web, Anolia, so Rosetta. So these are easy to use in ab initio structure of protein structure prediction. First steps in homology modeling. Actually, I take you directly to the website for predicting the model of a protein. But first, I am showing you a few more tools other than Swiss modeler for developing the structure of a protein. When we go with predicting the structure of a protein through computational methods, so if you see in NCBA GenPept, okay, We have around near 40 million sequences. We have around 40 million sequences. So 40 million, it comes to 400 lakhs, isn't it? Minimum 400 lakh sequences. If we have 400 lakh sequences, or how many structures we have in RCSP PDB website? Yesterday we have seen, isn't it? So it comes around 13 lakhs. Okay, so it comes around 13 lakhs. So we have a gap, isn't it? So the 3D structure information is not known for the 400 minus 13. If so, for these kind of a sequences, we can use the any computational prediction methods, can be homology modeling, threading, or ab initio. So here, whatever I have shown here, okay, this is a tool from Discovery Studio. So Discovery Studio is a commercial tool. In this tool, we have a program called Modeler, Discovery Studio Modeler. So in that, you have to feed just an amino acid sequence. And if you click submit, obviously you can able to predict homology modeling. I mean, predict the protein using homology modeling. Even we have separate tools for modeling the antibodies in Discovery Studio and similarly to model membrane proteins. So modeling the antibodies and membrane proteins is of course a complicated task. The online tools won't help. So for modeling this, obviously one should need the commercial tools like Discovery Studio, Scroid Inter, Tripos. So these Yasara, these are some of the tools we can use for modeling antibodies and membrane proteins because of complex structure. And whatever structure we have modeled using Discovery Studio Modeler, we can able to see the graphical view and aligning, how to align, how to change. If you want to mutate something, we can mutate in the sequence in the Discovery Studio itself, and we can able to go with predicting the protein structure for the mutated sequence. So building a 3D model using the next tool is the modeler. This is the second tool, modeler. Even through the modeler, we can able to model it. And a structure, whatever we modeled using modeler, you can find like this. So you can able to see DOP, score, PDF total energy, etc. in this modeler tool. Okay, this is how the result looks like. And one should see that there is one more plot generated called Ramachandran plot. What is Ramachandran plot? We'll be discussing in subsequent slides. So this is what we are going to try today. The one what we are going to try today is the Swiss model online tool. So our course in our course I'm going to teach you most of the freeware tools, can be an online tools or it can be an open source software, that is after downloading and installing in our desktop, we can open and we can practice. So I'll be teaching you most of the freeware tools, but I can also show you something like commercial softwares like MATLAB, etc. If I'm explaining to a particular concept. Okay. So, but other than MATLAB, you can also use any tool, whatever I'm going to explain from 1 to 14 sessions. Now, let me go ahead with the Swiss model. In the Swiss model, this is how a home page looks like. We have to simply click the start modeling. After clicking start modeling, just paste the amino acid sequence. Okay, the input what we require in homology modeling is just the target amino acid sequence. And the next input what we require is the template, okay, PDB file, that is 3D structure of a protein. One is the amino acid sequence, the other is the 3D structure of a template. For example, if you want to stitch a shirt based on your dimensions, okay, if you see Vanus and Allen Solly, these are popular brands. But the collar, if you like, from the Vanusen, and the handcuffs you like from the Allen Solly. So we can also model, I mean, we can also tailor the kind of shirt from these two brands. If you give these two shirts along with the bit piece, you can able to model the protein. I mean, you can design the shirt, isn't it? Similarly, if you are taking more than two templates. If you take more than two templates, we call that as threading. If you take a single target sequence and single template PDB, we call this as homology modeling. But I told you in the next case, that is, if I take two different brands and if I'm going to stitch, of course, we need a target amino acid sequence in the threading. But other than the target amino acid sequence, we have to require minimum of two templates. You can take as many of the templates, even 40 template PDB files you can take, and you can take which is the base from a part of a protein, that is a part of a motif of a protein. And if you start modeling it, we call this as threading. Okay, but whatever I explained, Swiss model works on homology modeling. A sequence, a structure is sufficient for modeling. So a structure, whatever we take, is a single template. Okay, and if you see this kind of a Swiss model, how the output looks like, I directly take you to the illustration, then I can come back to the PPT. So first I take you to the protein. So for downloading the protein, I suggest you to use UniProt, because if you go to the chain pept, you have so many kind of protein sequences related to the same protein. Then you will be confused between the isoforms. I told you, in the isoforms, some amino acids are missing. So if you want to know the correct length, so better go with this UniProt. So today I'm going to explain you with a protein called TRAP1. The TRAP1 also called as HSP75. You know, we don't have a structure for TRAP1 of mouse. So this is a UniProt ID, 6-letter ID, Q9CQCN, CQN1. First, I am getting inside the TRAP1, that is HSP75 of the mouse. So whatever we have, first I'm just going to the structure. If I click the structure, you can see we don't have a structure. Okay, if you have a structure, we have the PDB IDs. We just have the alpha fold, etc. But if you want to predict using your own kind of a computational prediction methods, better go with try with the sequence where the structure is not known. So I'll be taking the sequence from this by clicking the sequence. First, verify whether structure is available. If structure is available, better use that structure. Most of the structures of the proteins are complex with ligands. Delete the ligand and you can use it for docking or any other purposes. Now let me first take you the sequence, download the sequence. Click the download. The most happen is around 706 amino acid. Whatever we get now is a FASTA format. The first line is the identifier and the second line is the amino acid sequence. You remember I told you on yesterday that whatever we see in multiple lines or the wrapping of the text for user friendly. So we have this wrapping of text around 60 amino acids you can see in each and every lines. Okay, so totally 706 amino acids. The next thing what we have to go ahead with is Swiss model. Okay, so if you type simply Swiss model or Swiss model server, it takes you to the swissmodel.expacy.org. So whatever we have gone to through this, we can work with the homology modeling, isn't it? That's a model, that's a method of protein structure prediction. We can able to make it using this. So now we have to simply click start modeling for start. So we already have the FASTA format. Either paste the amino acid sequence or even if you paste the FASTA format, automatically it takes only the amino acid sequence alone. Okay, so one step we are making, just take a sequence and just drop. Okay, and if you want to directly build a structure, simply click build model. But if I want to get this because it takes some 5 minutes also to get the result. Okay, so better why can't we give safely with some email ID? I'm going to provide my email ID. I'm going to give a project title for this. Okay, and if you want to build directly you have to click build model. But there is also an intermediate step. If you want to search a particular template, like for example, if you have Alan Solly Van Heusen, the tailor is asking you either give one shirt, sir, then you will be giving Van Heusen, or some like the Alan Solly, then they can give the Alan Solly. So either one shirt template should be given and one bit piece can be given. From that, the shirt can be stitched, isn't it? So similarly, for selecting a template, selecting only one base template, okay, we can use this option. We get numerous templates. So if you don't want to go with this intermediate step, and if I want to directly build based on the algorithm present in the Swiss model, of course, in the Swiss model, you know how it takes a template. It takes a template which is having closely aligned with this amino acid sequence, which is more homologous with this sequence. Out of 706 amino acids of mice trap one, if this is more matching with the rat trap one, okay, rat that is Rattus norvegicus trap one, then it takes that rat trap one as the template, okay, and then it build the model automatically. So this is actually a built algorithm. So I'm simply going to click the build model. Okay, if you want to change anything, we can use that option also. It will take some time to generate the result. Meanwhile, I can go ahead with the PowerPoint. So, if I click the build model, okay, you'll be no, if I'm clicking a search for a template, you'll be getting these templates. So whatever you can see here, this is a first template. This is a first template, second template, third template. So whatever we want to select, that alone we can select. Okay, but we have skipped this step. I directly click the build models, isn't it? So we'll be getting this kind of an output page. In this output page, if you want to download the structure, actually we'll be getting two models, model 1 and model 2. So we can select downloading the models based on the homology percentage. So if you want to download, simply you have to click this and download. And there is also one more option for us called structure assessment. The structure assessment can directly take you to Ramachandran plot. You know, the Ramachandran is the father of bioinformatics in India. This Ramachandran, if you see his alma mater, various work. He worked in crystallography center from Madras University. Okay, so he is well-known scientist, well-known bioinformatician. He has developed a Ramachandran plot. You know what is the purpose of Ramachandran plot? The Ramachandran plot can able to identify the secondary structure elements of a protein sequence. Okay, it assesses the stereochemical quality of a protein. So to go through that, you have to click this option. Now let me take you to the next concept called threading. So threading, if you see, okay, here we try with all possible parents. I told you in homology modeling, we take only one best template PDB, but here we try with multiple PDBs. That's a major difference between homology modeling and threading. We optimize one model, even though we get two models, we take one model. Here, whatever we get in threading, we get many rough models, many. So 5 to 10 models you can get. We also have choices to select which is best. If my drug is not going to bound with the binding site of model 1, why can't we try with model 2? Why can't I try with model 3? So you have these kind of an options. But just because the homology modeling is going to try with a single parent, okay, the precision is less in homology modeling when compared to threading. In threading, we are using multiple parents, isn't it? So obviously, the precision is more. But one disadvantage is there in threading. The threading is too slow. If you want to get a result from homology modeling, especially through Swiss modeler, we can get a result within 5 minutes, 5 to 10 minutes, based on the size of protein. But whereas if you see the threading, Okay, I told you iTaser, a tool called iTaser. This can take a day, 24 hours to 72 hours for generating a result. So this is the disadvantage of a threading. And this is the homepage of the iTaser, janklabccmp.med.ums. So in Google, simply type the iTaser. You'll get this kind of a window. And there is also one disadvantage. So it's not a disadvantage. One should know how to use the ITSR first of all. So if you want to use the ITSR, you must register in ITSR. That is, you should have a login and password. Unless you provide the email, which is mandatory field, okay, and the ID, etc., you will be getting the result. Like however you worked in Swiss Modeler, similarly you have to paste the sequence here, copy and paste your amino acid sequence. And but if you have registered, you provide the registered email ID and click submit to get the result. I told you the advantages and disadvantages. So looking through that, we can proceed. Okay. And if you see this ITSR, I told you that whatever sequence we take, you're not going to predict with a single PDB, but with the multiple PDBs, isn't it? So might be if your protein is having some A, B, C, D, that is if 200 amino acids, you have 1 to 50 is the A motif, and 51 to 100 is B, and 101 to 150 and 151. If you have these four parts in the protein, so each and every part can also take nearly some 10 template PDBs. Here 10, here 10, here 10. Out of this 10, 10, 10, 10, okay, 1 to 50, it takes the best one template PDB to model. Okay, you know why we call this as a threading? In threading, we are going to have, in this example, if you see, we have four threads actually. Okay, the first thread, oh yeah, okay, hold on 1 minute. The first thread, okay, and if you go with the 51 to 100, you have a second thread. And then if you go with the not yellow, I take some other color. So this is 101 to 150, and this might be 151 to 200. So we have so many small threads generated, and we twine with each other to get the 1 to 200 amino acid protein. You got it? Instead of taking the entire sequence, I mean target sequence compared with the template PDB, it can able to take even a part of the target sequence, and it can able to whatever we generate a part of a protein, we can match and we can get a single PDB file. So we call this kind of a concept as threading. So twine the small threads to get a large thread. Okay. So this is a simple concept. Of course, for this, these are some of the steps you can find here. ITSR results. If you simply paste the sequence and if you go ahead with the structure prediction, we'll be getting the structures. As I mentioned earlier, we get many rough models. Model 1, model 2, model 3, model 4, model 5. We have plenty of models developed. Okay, so we can able to select any of the model and we can download and we can check with binding affinities. So next we'll be going to ab initio. So presently we discussed about homology modeling and threading. Now the third method called ab initio. What is ab initio? In ab initio, we predict the protein structure in by stepwise manner. Like if you predict the secondary structure from the sequence, then we can able to predict the tertiary structure. The tertiary structure can be predicted from secondary, and similarly quaternary. So stepwise, we'll be predicting the structure. Okay, now if you see this ab initio protein structure, of course, one should have a knowledge on bonded interactions and non-bonded interactions. So stepwise building from the atoms. Okay, we call this as an ab initio. So, okay, now we discuss the results. Now, can we check whether the results came? Yes, yes, we have received the results. So these are the results actually. These are the results of trap one. So actually can see is model one. Model one is having 78.53 percentage. On model two is having 88.49% sequence identity. Then which we can choose? The sequence identity should be more, isn't it? So I suggest you to download the model 2. To download the model 2, simply click download files, PDB format. So in this PDB format, whatever we download, okay, I'm going to save it in this model 2. And if you want to open this structure, so to open the structure, one can install Discovery Studio Visualizer from BioVia, which is a freeware. You know, the Discovery Studio 2023 visualizer is a user-friendly visualizer when compared to any other protein structure visualizers. So better download and install this BioVia Discovery Studio visualizer. So this is the structure of the mice trap one structure model from the homology modeling. So whatever structure we can see here, the blue color thing we can see, isn't it? That greenish blue. So this is what is the beta sheet. And whatever we see the coil, this is an alpha helix. Okay, this is an alpha helix beta sheets. Okay. Now, if you want to edit any content in the protein structure, use Notepad. Where if you even if you take a Microsoft Word, it may change the format. So if you want to change any coordinates, better edit using the Notepad or WordPad. So whatever 3D structure I've opened in Discovery Studio Visualizer, so if you see it in this notepad, so it will be like this. So whatever I told you from the protein crystal to the electron density maps to the final coordinate PDB. So this is the final coordinate PDB. And you know whatever the name of the protein we have given trap most that you can able to find it here. But if you have downloaded any structure from PDB, you can find the title. In the title itself, you can find the name of the protein. And you can see the journal information, who are the authors who have posted the protein structure, and you can see the remarks. So the most important thing one can able to see is the atoms. So you know that the amino acids are the building blocks of protein. And if you see this arginine, okay, the arginine, which is present in A chain of sixth position. So these are the atoms. 37 to 47 atoms are there in arginine amino acid. Like this, for the 706 amino acid, you can able to see the atoms. So here we don't have another kind of a coordinate called HETATM. So whatever you see in the first column, this is important to read the coordinates. To edit the coordinates, one should have a knowledge on how to edit the coordinates. So here we have only atoms. We don't have HETATM. If you have HETATM, we call that as other molecules, other atoms present in molecules. For example, in hemoglobin, iron molecule is present in the form of porphyrin. So iron molecule is present as HETATM. And similarly, if you see water molecules are also heteroatoms. Okay, so these are present in the form of. So if you want to delete any kind of heteroatoms, okay, so you can select the HETATM in the coordinate file. Okay, like the water molecules, whatever is there in the PDB file can hinder you while you go with the docking process. If you want to dock a small ligand molecule with a protein, you should not have an HETATM. But there are few HETATM molecules like iron in hemoglobin. That is most important to give a structure for hemoglobin. So you can't able to delete that kind of an iron. And similarly, manganese, magnesium. If it is a drug, no problem, you can delete it. But if it is a metal binding protein, we have to retain that. So now presently, whatever protein I downloaded on yesterday, xylose isomerase 1-XIH, I found the HET, HOH. You can see the HOH here. So I am deleting these coordinates. So while deleting the coordinates, just delete the HETM, that H2O. Then it can save like that. If SOR is something related to sorafenib, something like that, and if this tag is not needed, better to delete this also. So now, however, is saved. Okay, you can save it as such. So, how to edit the coordinates? I just teach you in the notepad. Okay, now let me go ahead with just I need 3 to 4 minutes to complete this today's session. So if you see whatever structure we predicted from computational methods like ab initio, homology modeling, and threading, we must compare with the gold standard technique proteins. That is the protein structures established from X-ray and NMR. So this is what we call it as a comparative modeling. This is what we call it as comparative modeling. Compare the computational prediction method proteins structures with the experimental structures. Okay. And if you see the Ramachandran plot, there is a lot of information in this Ramachandran plot. This is how a Ramachandran plot looks like in the Swiss modeler. So here one can able to see in this region we have plenty of beta sheets. And here you can see alpha right, that is bottom left. And this is alpha left helices. Here itself you can see pi helices, 310 helices, etc. If amino acids are lying somewhere here, okay, these amino acids are unfavored residues. But if amino acids are lying here, okay, in the shaded region, especially in the red color shaded region, we call this as favored residues. So according to Ramachandran plot, if you get favored residues, if we can get the favored residues above 80 percentage, in quality publications they mention like above 90 percentage. If the structure has more than 90% favored residues, that is you have these residues placed only in the desired locations, we use this protein for the docking and simulation. We can presume that protein quality is good, and we can take it for any other computer-aided drug design applications. And even here in beta sheet, you can see the parallel, anti-parallel, so other kind of locations you can see. Okay. So one more thing to assess the protein quality other than Ramachandran plot is RMST. Root means square deviation. So whatever experimental structure you can see, these are the atoms might be 1, 2, 3, 4, 5. These are even an amino acid molecule atoms. Okay, and if you see this is determined through X-ray or NMR and predicted is from the homology modeling threading, something like that. If you have a small gap, I mean, if you have a small distance between the center of the radius of the atom of an experimental with the predicted, then if RMST value is low, then the prediction is more appropriate. If RMST value is high, then it is whatever predicted is having some kind of a bias. Okay, this is how we can able to find. CASP is one of the tool to predict and assess the quality of a protein. Okay, and we can also assess through these tools like Verify 3D, ProCheck, etc. Okay, so today we have completed like how to model the target, how to assess the target, and how to validate the targets. Okay, so today I've completed. Can you repeat X-ray PDB? So I think I've repeated. If you want, you can ask me once again. I've completed today's session. If you have any doubts, you can ask me. Thank you. Thank you. And tomorrow, we'll be discussing on methods of drug design. Okay. So in methods of drug design, we have plenty of methods. How can we get recording? Yes, you'll be receiving in your registered email ID. So whatever email IDs you have provided, you'll be receiving that on that day itself. Even yesterday, I've sent you the video. If you didn't receive, send me your email ID to my WhatsApp number so that I can send you. Thank you. Any other doubts you have, participants, can ask me. If no doubts, thank you very much for your patience and interaction. Tomorrow, we'll be discussing on a new topic. So, thank you. Have a great day to you all. Bye.